Hello everyone, welcome to my tutorial on cellular respiration. Here in part 1 is just give you an overview of the various steps and what their purpose is, and then parts 2, 3, 4 will go into more detail into each parts. So this is an overview, and what we're doing is we're taking a of glucose. Now this diagram is only showing you the carbon, but realize there's oxygens and attached to it. And we're going to take that C6H06 and we're going to convert that into 6 ATP. And if you remember last semester when we talked about ATP, what we were talking about was that ATP is sort of the currency of life. And even though glucose is an energy molecule, it's too big of a molecule to be used effectively by the cell. It'd be analogous to you going to 7-Eleven with a $1,000 bill to buy a Slurpee. Even though the $1,000 bill is currency, it's too big of a currency used by the store. So what the store would ask you to do is to take that $1,000 in and convert it into $20 bills. You would have $20 bills, and then you could use the $20 bill to buy the Slurpee. Well, your body's doing the same thing, or you're saying that they're converting the glucose molecule, the large unit of energy that's too big to be utilized by the cell, in smaller units, 36 ATP units. The process occurs in several steps, the first one being glycolysis, and then an intermediate reaction right here, followed by what's called the Krebs cycle, and then where most of the energy is actually made is here in the electron transport. In fact, 30 of the 36 ATPs are made in this last step. The Krebs cycle produces 2 ATP, and this also produces 2 ATP. And although these steps here don't produce as much ATP, what they do produce are hydrogens that are going to be taken to the electron transport system down here, where they're going to be utilized in the same way that plants use the hydrogen in photosynthesis, in the light reaction of photosynthesis. Remember back to photosynthesis, what the plant did was it used water, got rid of oxygen, and used the hydrogen in the water as a source of electrons to power the light reaction. And also to use after the hydrogen had lost its electron, a proton, and remember that it would concentrate those protons on one side of the thylakoid membrane, build up a high concentration, where then they would go from high to low through ATP synthetase to produce the ATP in the light reaction. In cell respiration, we're going to use, instead of water, we're going to use glucose. And just like the plant got rid of the oxygen, the mitochondria is going to get rid of the carbon and the oxygen, and we're going to utilize the 12 hydrogens, same way that the plant used the hydrogens, as a source of electrons to power the electron transport system, and concentrate protons on of a membrane and force them to go through ATP synthase to produce the ATP. So these steps, glycolysis, the intermediate reaction, and the Krebs cycle, are all designed to get rid of the carbon and the oxygen and liberate the hydrogens, taking them to the last step where most of the ATP are going to be produced. So we'll go over each of these steps in more detail in later parts, but it starts with our 6-carbon glucose molecule and then splits that in 2, just like that, with 2 molecules of what's called pyruvate. And this part, glycolysis, occurs in the cytosol. So even if you're a prokaryote and you don't have a mitochondrion in your cell, you're going to be able to produce energy through glycolysis, although you're not going to get a whole lot of energy, only 2 ATPs. So the pyruvate then goes into the mitochondrion, and so you have 2 3-carbon pyruvates. What we want to do is reduce them down to 2 carbon molecules. And we do that by removing a carbon dioxide. So if we had 2 3-carbon molecules, each one loses a carbon dioxide, then we end up with 2 2-carbon molecules to go into the Krebs cycle, like this. Similar to photosynthesis, remember in photosynthesis we had the 5-carbon ribulose diphosphate, which would pick up a single carbon in the carbon dioxide, form the 6-carbon molecule. In cell respiration, rather than a 5-carbon resident molecule, we have a 4-carbon oxaloacetate that's going to pick the 2-carbon molecule here to form a 6-carbon molecule. At any rate, this 2-carbon molecule is then going to go through the Krebs, since there's 2 of them, it will go through twice, and we're going to produce 4 carbon dioxides. And by the end of these 2 steps, all of the carbon, all of the oxygen that were in the original glucose have been taken into your circulatory system, where they're taken to your lungs and then exhaled into the environment. And what's left behind are the hydrogens. In photosynthesis, remember you had NADP as the hydrogen carrier. Well, in cell respiration, we have a close cousin of NADP called NAD. And it's a charged molecule, and so it's looking for a negative hydrogen, and to do that, it has to be carrying 2 electrons here. So imagine D plus is going to come up and pick up a hydrogen ion carrying 2 electrons, and those electrons and the hydrogen are going to be carried to the transport system. Same thing is happening coming out of the Krebs cycle. So all that's left that goes into the electron transport system is the hydrogen, electrons carried by the hydrogen, and this is where most of the ATP is going to be produced. See you in part 2.